It's nice to be with you again. We are here in another session in microbiology, and in this session, we are going to discuss microbial growth. Under microbial growth, we have to discuss bacterial nutrition, or what are the requirements of a certain bacteria in order to grow. The following are the factors that affect bacterial growth: temperature, pH, osmotic pressure, oxygen classes. Let us take all this one by one. So you have cardinal temperatures, the minimum temperature, those that require optimum temperature, and maximum temperature. Temperature is one of the major environmental factor that controls microbial growth. Here is the graph that shows to you the microbial growth. So if you can see this green line here, it shows to you the minimum temperature that requires for the microorganism to grow, and this is also the optimum, and this is the maximum. The difference between the optimum and the maximum is the optimum is the desired temperature in which the highest peak of microbial growth is present. For the maximum temperature, this is the highest temperature in which microorganism could grow, but it does not mean that this is the peak of the microbial growth. It is the optimum temperature that provides the bacterial growth. Now, let us try to look and differentiate the different temperatures for microorganism. We have the minimum temperature, optimum temperature, maximum temperature. As we have made mention, minimum temperature is the temperature below which growth ceases, or it is the lowest temperature at which microorganism will grow. And then in optimum temperature, the keyword here is the growth rate is the fastest. This is the temperature in which it is very favorable for the growth or increase in number of bacterial cells. For maximum temperature, it is the highest temperature at which microorganism will grow. There are different classifications of microorganisms according to temperature requirement. So, the temperatures that survive at low temperature, that is 4 degrees centigrade, are called the psychrophiles. Although there are still some other microorganisms that could even survive lower than 0 degrees centigrade. And these are now the things or the microorganisms that um, some microbiologists are interested with, especially those who are interested in the history of microorganism. And then we have also the mesophiles. The mesophiles tribes something that is within the body temperature or above the body temperature, that is 39. And then we have the thermophiles, which starts um, to increase its number in the simmering temperature of water. So they are called the thermophiles. And then we have also the hyperthermophiles, those that could survive at high temperature, that is near to boiling water temperature. And they could also survive as high as 106 degrees centigrade. Example of psychrophiles or that lives low temperature are the Polaromonas baculata. For mesophiles, of course, the very popular Escherichia coli. They reside in a temperature in which the body temperature is beyond normal. The normal body temperature is 37. If you have fever, you have a temperature of 39 degrees centigrade. And then you have also Bacillus Styrothermophilus and Thermococcus celer and Pyrolobus formari are examples of thermophiles. For the other two, they are called hypothermophiles. So let us try to look into the microorganisms in details. The mesopile survives from temperature 20 to 45 degrees centigrade, and they could even live within warm-blooded animals and also likewise terrestrial and aquatic environments. Um, these are also usually found in the near near rivers or near in icy areas. And then you have the psychrophiles. They are the cold-loving microorganisms, and they live in extremely cold environment. Then thermophiles, they carry with them their life within 45 to 80 degrees centigrade. And higher than 80 degrees centigrade are the hyperthermophiles. So these are examples of microorganisms also according to temperature and according to the presence of light. So at minimum temperature, optimum temperature, maximum temperature, these are the list of non-photosynthetic prokaryotes, and these are the photosynthetic prokaryotes. What is interesting here is these microorganisms are not only classified according to temperature but also to the presence of light. So non-photosynthetic prokaryotes are the microorganisms that do not require sunlight, but for the photosynthetic, they require light. Not necessarily sunlight, but also other artificial lights that could be used in the laboratory. And then these are also the microorganism that thrives in higher living animals um, vis-a-vis the temperature. So you can see here that a microorganism could survive within the within and outside the parts of fish, insects, and crustaceans. The same true with other living organisms like plants and eukaryotic organisms. So you have also here the list of some microorganisms living in other temperature higher than uh, room temperature. Now let us go into pH. pH is the alkalinity or basicity of an environment. Most organisms best grow in near neutral, that is 6 to 8. Although there are some microorganisms that loves highly acidic environment and basic environment. The microorganisms that loves acidophiles like your Helicobacter pylori, Thiobacillus thiooxidans are acidophiles. They grow best at low temperature or they like acidic environment. The alkaliphiles are organisms that grow best at high pH or basic. Example of which is Vibrio cholera. And then you have also the neutrophiles. The neutrophiles lives within the pH 6 to 8, and usually most of them are pathogenic bacteria. And then these are the classifications also by pH requirement and based on um, place. So the acidophiles stays here from 0 to 6, and then the neutrophiles belongs to this pH and could be found in pure water even, and then the alkylophiles. Osmotic pressure or the salinity or presence of solute within the water solution. So these are the classifications based on um, salinity. The osmophiles lives at high. The osmotolerant, the solute is also high. And then the halophiles in high salt. The halotolerant They could tolerate high salt. Barophiles and the barotolerant for high pressure. Okay. These are examples of microorganisms that could grow based on water activity. And then halophiles and related organisms. Um, Osmotic effects are interest of many microorganisms that are residents or host habitats belong to high salt environments. So you have examples here, the Halobacterium and Dunaliella. They could live uh, in seawater. And then you have also halotorrellant. They could tolerate some reduction in water activity, but they grow best in the absence of added solute. So meaning to say, they also could grow in the environment with high concentration of salt, or the salt that I'm referring to are the salts that are defined in your chemistry. Then you have xerophiles. They grow in dry environments or low water content. So here are some examples. The halotolerant belongs or is represented by the green line, and then the halophiles, the brown, and then you have here the peach color for the extreme halophiles. Now let's go to bacterial nutrition. Nutrition, of course, provides the microorganisms to grow, and they are either classified as macronutrients or micronutrients. The main macronutrients for microorganisms includes the carbon. So this includes the carbohydrates, protein, and fat. And then another is nitrogen, 12% of the dry weight. So in agriculture, the nitrogen fixing bacteria are very important because they convert nitrogen from air into nitrogen compounds that are essential in the growth of plants. So that those bacteria are called nitrogen fixing bacteria or nitrogen containing bacteria. They have the capability to use NH3 and also NO3. So you have here the chemical illustration, how the N2 in a triple bond, how the N2 is converted by enzymatic activity with the help of nitrogen fixing bacteria, plus of course the reaction of adenosine triphosphate to form the ammonia. The ammonia serves as the nutrient for plants, or what we call fertilizers. So another classification is their ability to work with light and also chemicals. We have the photoautotrophs for those that requires light, but those who do not require lights but make use of chemicals or other nutrients without using light for conversion of food are called chemoautotrophs. And then for those who made use of carbon dioxide, they are called autotrophs. So the light the light needing bacteria that requires carbon dioxide are the photoautotrophs. And then for those that requires chemical compounds, they are called chemoautotrophs. But those that make use of organic compound, not carbon dioxide, are called heterotrophs. So if they need light, they are the photoautotrophs. and the other one is chemotrophs. Other nutrients includes minerals, phosphate, sulfur, potassium, magnesium, calcium, sodium, iron. Iron plays a major important role in respiration, and sometimes there are some microorganisms in which their appendages um, were given color because of the presence of iron that is colored brick red or orange or red or brown in color. Then we have siderophores. They're iron binding agents that cell produce obtain iron from various insoluble materials. These are the enumeration of macronutrients in nature and used also in the laboratory through your culture media. And then these are also the micronutrients present in some of the foods given to microorganism in the laboratory, or even they could be obtained in nature. So these are some organic compounds that they also need in the form of vitamins. And then let us try to look into what happens in the laboratory if oxygen is present. If if the aerobes is obligate, they require oxygen to grow. If facultative, they could live with or without oxygen. Microaerophiles, kindly correct this spelling. Microaerophiles requires the level of oxygen. So in this manner, the presence of oxygen uh, is not possible here. So the microorganism goes down in order to avoid the exposed area with oxygen. But in this case, they go and float at the surface looking for a supply of oxygen. And these are facultative, they could live with or without oxygen. These are also illustration of with or without oxygen. Okay. So these are still discussion for obligate aerobes and obligate anaerobes. If you say obligate, they strictly do not require oxygen. If they are obligate aerobes, the presence of oxygen will not allow them to grow. This is very true for the bacteria used in making wine. And then obligate anaerobes, they cannot survive in oxygen. So there are also some winemakers that covers their barrel or their bottle in order for oxygen not to grow. But there are those that need fermentation that requires oxygen. And then facultative anaerobes, they grow better in oxygen. Aerotolerant, they don't care about oxygen. So microaerofoils, they require low level of oxygen. So this is the summary. And then you have here the different graphical illustration of microbial growth. So the microbial growth divides by a binary fission. So the microbial growth could be slow but increasing. It could also be very fast in terms of growth. So this is the time, this is the number of microorganisms. So in this case, in letter B, the, the, the symptoms of the diseases could easily be seen as compared here because the number of microbial growth is faster as compared here. Okay, and there is also cases whereby microorganisms could already be in a thousand-fold within just a short period of time. This is just a review of binary fission as an illustration to you. I am sure you all of you knows what is binary fission. So this is the graphical presentation as to what is binary fission all about. And then you have the growth curve. There are three stations of the growth curve: the lag phase, the log phase, stationary phase, and the dead phase. The lag phase is where the microorganisms are trying to grow. This is the recovery period for them to assemble themselves. And then for the log, there is already an equal amount of death and equal amount of growth. The microorganisms are growing very rapidly. In the stationary phase, the growth rate is equal to the death rate, so it is stationary. And then as you pass the stationary phase, it could lead to death rate whereby the growth is getting lower and lower. In the laboratory, there are different ways to measure the microbial growth. One is count colony on plate. One is directly microscopic count. We have also flow cytometry and turbidity. In the laboratory, you will just be simply doing the count colonies on plate and observe liquids in terms of turbidity, but just simply in qualitative form. You will not be doing these things because this requires electron microscope or microscope with higher magnification. These are also some illustrations that you will be supposed to be doing in the lab to show to you the effect of oxygen to microorganisms. So these are also some illustration to measure growth.